Thank you for having me here. My name is Parul Sharma. I am a bioinformatics scientist at Emory University. And today I'm going to be talking about azithromycin resistance in Chlamydia trachomatis, specifically what large-scale genomics can and cannot tell us about resistance in this pathogen. So to start things off with some context, Chlamydia trachomatis is an obligate intracellular bacterium and the most common bacterial sexually transmitted infection worldwide. Clinically, it also causes urogenital and anorectal infections, along with lymphogranuloma viridium, which is a more invasive infection that affects the lymph nodes, and as well as ocular trachoma, which remains a leading infectious cause of blindness globally. So treatment typically involves doxycycline or azithromycin as the antibiotics of choice. Importantly, azithromycin has also been used extensively in mass drug administration campaigns, particularly for trachoma. Now, one major challenge with Chlamydia trachomatis is that the most infections are asymptomatic, with over 70% in females and about half of the males being asymptomatic, which complicates detections, treatment, and also interpretation of treatment failures. So at the genomic level, Chlamydia trachomatis is structured into 4 major lineages, which are clearly resolved at this core genome phylogeny. Now, these lineages are associated with different disease phenotypes. Some of them I've already talked about. So the LGV, which stands for lymphogranuloma viridium. Then there are non-prevalent urogenital and anorectal infections, and then prevalent urogenital and anorectal infections. And then there are ocular strains. So these are all highlighted in different colors of the different 4 major clades in this tree. And on this tree, I've also annotated OMP-A types, which is a genotyping scheme usually used to identify Chlamydia trachomatis. And you can see that the LGV and ocular lineages have fairly distinct OMP-A types, but whereas there are some overlap between the NP-UA and the PUA clades. Another technique to genotype Chlamydia trachomatis is MLST sequence typing. And in the outer ring of this tree, I've highlighted the MLST for all these strains. But an important point I need to point is that MLST doesn't reliably capture the structure always, because many of the genomes with these gaps here, you can see, remain unassigned, which is why whole genome approaches are really essential here. Now, over the past decade, several studies have reported azithromycin treatment failures in trachomatis. And what's interesting is that in these failures are often better explained by the detection of resistance-associated mutations, along with the traditional MIC measurements. And at the same time, there have been a growing effort to catalog resistance genes and mutations for chlamydia, largely by borrowing markers from other bacterial species as well. And that's something we want to critically evaluate. So overall, the aim of this study was to examine the azithromycin resistance in globally circulating Chlamydia trachomatis strains using large-scale comparative genomics. And specifically, we wanted to build a comprehensive, high-quality genome dataset and then evaluate resistance by systematically looking at candidate genes along with those mutations. So we aggregated genomes from multiple sources. So these included high-quality assembled genomes from NCBI, and then also assembled SRAs from all the bacteria database, reference genomes that we had sequenced in the lab, along with some sequencing data that we collected from infected patients for other studies. In total, that gave us about 1,714 genomes, and after quality filtration, selecting for high completeness and low contamination, we retained about 1,349 high-quality assembled genomes. So for the resistance, we pulled from public databases, such as the NCBI AMR database and CARDS databases. And we also complemented that with extensive literature survey identifying mutations that have not been reported in these databases. So we focused our attention mainly on three candidate loci, and these were often implicated in macrolide resistance. These included the 23S rRNA sequence, the RPLV gene, and the RPLD gene. Now, this slide shows one of the key results from our study. And so to break it down, we have the core genome phylogeny tree of all the strains that we used in the data set, which were 1,349 genomes. So we have that on the left here. And then on the right, we have this heat map of sorts, which lists all the substitutions we identified in the RPLV gene, along with whether or not they were present in that particular strain. So across the data set, we identified 16 total mutations in the RPLV gene, but only 3 of them have been previously linked to azithromycin resistance, and these are the ones that are highlighted in this red box. And what really stands out in this graph is the lineage specificity for these three mutations. So what you can see is that all the LGV strains uh, carried the G52S and the V77 mutations, and nearly all, so about 99% of the prevalent strains had all the three mutations. So these mutations are definitely not rare or sporadic events, but they deeply embedded within these specific lineages. And it could be that these two major disease-causing lineages are spreading because of this, and that's why they're evading antibiotics. So to understand whether these mutations arose in response to azithromycin use, we performed a time-scaled analysis. So there is a phylogenetic tree that I created using BEAST. And for context here, I want to say that azithromycin was first discovered in 1980 and approved for medical use only after 1988. However, the three oldest strains in our study that we were able to find tell a completely different story. So one of the oldest strains that we found was a 1959 ocular strain from Taiwan, which had no mutations. And then another one from the same year, which belonged to the prevalent clade, carried the triple mutations that have been previously linked with azithromycin resistance. And then a 1967 strain that was from the LGV clade possessed the double mutations that I was talking about. So along with this and the time scaling, this indicates that these putative mutations definitely predate the widespread use of azithromycin by decades. And next, we wanted to look at the 23S rRNA mutations that are commonly annotated as macrolide resistance markers from public databases. So like I previously said, I found mutations that were listed in NCBI AMR database as well as CARD. And these are the list of the different mutations that I was able to curate. And we found several mutations, but most importantly, there were 4 of them that were present in nearly all the genomes in our data set, with frequencies up to 98 to 99% of the strains containing that mutation. And as you can see, these mutations originate from resistance annotations in other bacterial species, such as Neisseria or E. coli. But in Chlamydia, they appear to be the baseline species variation rather than any indicators of resistance. And this kind of analysis suggests that these variants represent fixed or highly conserved positions rather than any markers of acquired resistance. So next, this is an overview of a similar analysis, now looking at the 23S mutations that were detected and all the mutations detected in the RPLD gene mapped onto the phylogeny as previously. And again, we see is strong lineage specificity rather than patterns consistent with recent drug selection. And there's no clear signal of convergent evolution across lineages, but that would suggest widespread acquisition of azithromycin resistance. And again, clear lineage-specific patterns for both these gene sequences. So overall, what this suggests, just to summarize in the study, is that first, well, putative resistance mutations in Chlamydia trachomatis clearly predate the clinical introduction of azithromycin. Secondly, there is a strong lineage specificity, which is more consistent with neutral evolutionary processes rather than antibiotic-driven selection. And third, that azithromycin treatment failure is probably multifactorial, involving other genetic effects, host or physiological factors, and these could also be the treatment failures that we see could also be from reinfection from an untreated partner or inadequate drug treatment or non-compliance with medications. And finally, the point that I want to make is that resistant markers cannot be assumed to be transferable across species. And studies like this can be really helpful to refine targets that are worthy for testing experimentally for true clinical relevance. And with that, I want to acknowledge, so the group here that we work with, we call ourselves the ClamBig group, and we have collaborators from UCSF along with our team at Emory, who all have helped me envision this and make it to fruition. I also forgot to mention that this is a published study. We recently got it published here, and if you want to read more about it, you can refer to this link. And if you have any questions, I'll be happy to take them now.